Hi everybody, I'm Dave Tabb, nice to meet you all. We'll be working together this week on a program that we hope is going to be sponsored by Diplomics. Diplomics is a program run by the Department of Science and Innovation. This year, Department of Science and Technology has been joined by Department of Higher Ed to make this DSI organization. And so there's a much stronger emphasis now that Diplomics take on an educational role, and this gives us the opportunity to teach much more broadly this material. I normally teach a fair bit over at the University of the Western Cape, and of course at my home campus, the Tigerberg campus of Stellenbosch University. But I'm really glad to get to talk to people even more broadly than that. One of the key messages that we want to convey with this bioinformatics training is that you can gain a very powerful, very marketable skill set to be ready to do graduate training or postdocing or whatever at great universities. You have passed a lot of tests to get to this point in your career, and I'm hopeful that the addition of bioinformatics training skills will get you to a place where you can go to even better opportunities in the future. Bioinformatics is probably one of the most mobile of skill sets that everyone in biology now needs these skills. And it gives you a much better opportunity to move to better and better labs by developing these skills. So I'm glad that you're here today. I'm glad you made it a priority to be here. And through the course of this week, we're going to cover a lot of material that is very foundational in the space of bioinformatics. Okay. So we're going to be talking about some of the most basic of technologies associated with bioinformatics. There's a world of people out there who believe that bioinformatics is all about sequencing stuff and about alignment, sequence alignment stuff. So these are really foundational issues, and I think that most people will find that a lot of bioinformatics topics don't really come naturally if they don't have background in the concepts of DNA sequencing, sequence mapping, and sequence assembly. And all of that is what we're going to handle this morning. So we're going to start with a little overview of sequencing chemistry. How many folks here would feel comfortable right now explaining how massively parallel or next-gen sequencers work? A lot of people, frankly, in biology treat them as black boxes, but I hope by the end of today you're going to have a better sense of how the old school sequencers compare to these new ones that we use. You know, if you throw your samples at an Illumina, how do you get data out of that? We're going to be talking about base calling errors. Essentially, how do we transform the signals that come from the sequencing experiment into letters, and how much confidence can we place on the individual letters of a sequence? We're going to talk about mapping versus assembly, which is one of the key differences in how we deal with the data that come back from sequencers. And we'll talk about the Burroughs-Wheeler algorithm and suffix arrays, kind of a complex topic, but one that's important to think about. And then we'll talk about assembly, which is in some ways a mind-blowingly difficult thing, but which is in fact something that we're capable of processing on a desktop computer. Along the way, we'll talk about a few file formats that are rather important that everyone should be familiar with. Those will be FASTQ, SAM and BAM, and FASTA. All right. I think everyone should be reasonably familiar at this point with the fine structure of DNA. We have phosphates alternating with sugars. In the case of RNA, these sugars are 5-carbon riboses. In the case of DNA, deoxyribose instead. We have nitrogen bases that are attached to these sugars that point inward. And remember that we have a sense of orientation with DNA, that you have a 5' end and a 3' end. Does everyone know that the 5' and 3' ends are named that based on the carbons in these sugars? Right, so you can see that this is carbon 1, 2, 3, 4, and 5. So the 5' end relates to that carbon number 5, and then the 3' end relates to this 3. So we see that we're always building in the direction of 3'. So the polarity that we have on the sequence and so on relates to that. I want to point out as well that some of the technologies that really dominated sequencing for forever were built around the idea that this attachment point is available for the addition of another nucleotide. When we use Terminator sequencing, we are adding nucleotides to these sequences that block the further addition of additional nucleotides. So this is one of the ways that these that a lot of the sequencing methods we use create complementary sequences that don't cover the entire length, but stop at, say, the 30th position, or the 31st, or the 32nd. It's because they've incorporated a residue that blocks further additions. We have kind of a diagram of that here. You can imagine that you have this piece of DNA at the top that we want to sequence. So our process for doing so is going to involve building complementary sequences that cover different extensions of that. So you could imagine that the very shortest possible one you could incorporate would be just that very first nucleotide, the T. You might have another one that extends a little further, that includes just the last two T's, or the GTT, and so on. We're not directly controlling which strand gets a dideoxynucleotide, right? Our solution for performing this sequencing reaction includes both ordinary nucleotides and dideoxynucleotides. If by chance a dideoxynucleotide is incorporated, that prevents further extension of that complementary sequence. So we have a whole range of different products that are created in complement to the original template sequence. Now, I imagine at this point, you picked up this material back when you were undergrads taking biochem class, but it's important, I think, to remember that it's still very much live information. I would also point out that this is a rather different mechanism than what is typically used by machines like Illumina sequencers and so on. We frequently see a strategy where we're detecting the arrival of each nucleotide by its by a light that's emitted by the incorporation of a new nucleotide. So the upshot of this is this Terminator sequencing gives us a whole series of complementary DNA pieces that contain varying numbers of nucleotides in that complementary sequence. Now, back in the old days, we would use a separation that would allow us to measure essentially the single the different complementary sequences that have been differentiated by just one nucleotide from each other. We'd be able to measure, basically interrogate which dye was incorporated in this complementary sequence and what is its size relative to all the other complementary sequences. Now, that might seem a little complex, so I want to try, we're going to talk about electropherograms in a little while that represent all of these different color traces that came back from the complementary sequences. Now, a lot of times, you'll find this called next-gen sequencing. But I would also point out that we've been calling it next-generation sequencing since I was a postdoc. And that's a very long time. At what point does next-gen get supplanted by the next-gen? It's kind of a bad name, right? So I'm going to refer to this as massively parallel sequencing instead. Massively parallel sequencing represents the case where instead of attempting to devote an entire capillary electrophoresis column or an entire gel lane to one particular template we're going to sequence, massively parallel sequencing is focused on sequencing millions of different templates all at the same time. So although the old school sequencers, like the ones you get from ABI back in the day, you might have had 384 capillaries. What that meant is you could sequence 384 different template sequences all at the same time. That's child's play compared to what you can do on your basic MySeq today. So we have lots of options for doing this, but all of these technologies really build upon the ability to sequence many templates all at once. Okay, so a lot of times these are working via the measurement of incorporation. That if you allow an A to attach to the growing strand, you're going to get a flash of color that represents A. If you allow C to be incorporated, you're going to get a flash corresponding to C, and so on. So this these approaches are not uniform. Like if I'm talking about a PacBio sequencer, an Illumina, or whomever, or the thermo-ion torrent, they're all going to have slightly different technologies. But almost all of these give us the ability to create bazillions of copies of a particular template sequence, all located in the same very small location on this photocell that we're working within. So I may have the possibility of measuring up to 5 million different templates. I have 5 million different individual strands of DNA that have been sorted to a particular location in this space, and we've amplified to create a bazillion copies of it at that location, which gives us enough light that we can sequence that particular molecule. Okay, so we need to separate the templates, we need to fix them on a substrate. Typically we have some means by which we amplify, we create many, many copies of that template at that location, and then we're going to detect the incorporation of each base at each location. So I want you to imagine that each of these little blue plates is our photocell, right? Now, in reality, we're going to have millions of spots here, not just 4. But this little frame-by-frame cartoon is going to help us understand how we can sequence multiple things at the same time. So I want you to imagine that we're effectively working through a little cartoon. At the first time point, we incorporate nucleotides for each of the templates at each of the locations. And we get a little spot of green up here, a little spot of orange, or yellow over here, red over here, and blue over here. That tells us that the first nucleotide at this location is a T. The first nucleotide at this location is a G, and so on. When we incorporate this second nucleotide, we see that we get a flash of red up at the top rather than green, which is what we saw in the first place, or a blue at the left rather than a yellow at the left. The software remembers over time what color flashes have happened in what order. As a result, we can read this particular spot, the templates that are here at the left, as first incorporating a G, then incorporating a C, then incorporating a T, then a G, then an A. So the software infers that the sequence associated with this particular position in the photocell is GCTGA. So this is a little like watching a cartoon unfold. You know, you have like the first drawn cell, and your eye perceives motion because the second cell has the same objects just moved slightly, right? So in the same way, each cell of each incorporation step is going to produce a different color that's going to allow us to discern what sequence is present at that position. Okay. Now, I've already broken out a few of these terms, but I want to try to define some of them as well to make this a little more straightforward. So typically, we talk about the DNA that we want to sequence, the part that we're going to make a complementary sequence to, as a template. So that's a piece of DNA that we're going to sequence. Frequently, we are using something like paired end sequencing. We'll talk about that a little later in the slides. And this is a case where we have a long piece of DNA, maybe thousands of nucleotides, and the parts that we're going to sequence, we're not going to sequence the whole thing, we're just going to sequence the very ends, like the 5' end and the 3' end, we're going to sequence just those patches. So for that, we have a longer piece of DNA, often called an insert, from the days when people did a lot of like bacteriophage libraries and so on, often called an insert in that case. Now, we have as an input to the sequencer a template DNA piece, and the output is a read. That's the sequencer's interpretation of what it saw for that piece of DNA. We're going to talk about what these reads look like and the kind of information associated with them as we move through this first half. Frequently, you'll hear the term shotgun sequencing. Now, whole genome shotgun is quite a different strategy than the kind of sequencing we were doing back when I was in grad school. See, I was in grad school in the late 1990s, and we my lab was associated with the genome sequencing facility at the University of Washington. They had a very different notion about how to do sequencing back then, and the reason was sequencing was friggin' expensive. You couldn't just sequence everything you wanted to do. You could pick and choose what things you wanted to sequence, and you would get your 384 templates sequenced by one run of the instrument. And these were not cheap instruments, right? These were very expensive instruments. So the idea was sequencing is precious and expensive, so we're only going to sequence those pieces of DNA that help us to cover regions of the chromosome that we haven't already observed. That's a very different perspective than what we do now. So if you're doing whole genome shotgun, you have a lot less control over which pieces of DNA are going to get sequenced. But and generally speaking, you're dealing with sequencing reads that are much shorter than in the old days as well. So what you're hoping to do is effectively work with an old quote from, I believe, Stalin, who said, "Quantity has a quality all its own." That the old day sequencers, the old Sanger sequencers and so on, would give us very few sequences, but they were longer and they had better quality in general. Now, with whole genome shotgun, we assume that any deficiencies in our sequencing reads, they might be rather short because of the technology we're using, and they might have lower base pair quality. That because we're sequencing bazillions of them, that's going to be okay. We'll talk about how that plays out as we move into assembly. And finally, we have fold coverage. Fold coverage. You will find that if you go poking through the taxonomy at NCBI, the National Center for Biotechnology Information, that almost any animal you can name has had somebody try to do some sort of sequencing associated with it. We sequence everything. However, not all sequencing is done at equal quality. For example, if you were to look at an individual nucleotide in the sequence, the genome sequence reported for E. coli, you're probably going to have a higher confidence associated with it than somebody who has detected, say, cave bear DNA in a coprolite, right? They're sequencing at high quality and sequencing at low. One of the ways that we characterize the quality of sequence we have for a species is by fold coverage. So if you are working with a fungus that has kind of a very loose bit of knowledge about it, it was found in this particular sample, but we don't really have anything else in a similar genus and so on, and we've sequenced it at 2x coverage, it means if its chromosome is X, if its genome is X nucleotides long, we've produced only 2x nucleotides in sequencing reads. That's not very good, because if you do only 2x coverage for an organism, you are not going to have a complete end-to-end sequence for its chromosomes. Generally speaking, someone's going to have upwards of 10x coverage before they have what we'll soon call a scaffold that can cover a whole chromosome. So fold coverage is one of these terms of art that we use to describe how complete is the sequencing that's been done in this organism. So a lot of people will say, gosh, if we have massively parallel sequencers, if we have next-gen sequencers, why would we bother with the old school Sanger strategy? So let's talk about the old versus the new. Generally speaking, when we talk about capillary electrophoresis, fluorescent tag, automated sequencers, the kinds of things that completed the Human Genome Project back in the late 1990s and early 2000s, we would see that an individual template would be sequenced to a length of about 600 base pairs. 600 base pairs. Today, it's very frequently the case that our next-gen sequencers are cranking out about 150 base pairs instead. Now, some of them can go much, much longer than that. There's a lot of reason to think that when you're sequencing a novel organism, that doing so with a combination of very long sequences and a massive number of very short sequences is actually a nice gain, because the very long sequences help you with scaffolding, whereas the very short sequences, by the very large numbers, ensure that you have the nucleotide sequences correct. So in general, when we talk about stuff like Illumina sequencers, we're talking about relatively short reads. Generally speaking, we would have a greater sense of accuracy about individual reads in the old school Sanger sequencers. But, as I said, there's a quality to having very deep coverage that results from having lots and lots of templates sequenced. So by playing those off of each other, you're able to compensate for the relatively high error rate associated with these shorter reads. Because we're able to sequence from millions of reads rather than tens or maybe hundreds, maybe a couple hundreds templates, we find that massively parallel sequencing gives us oodles more information to work from. It also means, however, that we have a considerably more difficult time storing all the data that come back from the sequencer, and quite a lot more bioinformatic challenge in making sense of them too. So there's a lot of weight placed on bioinformatics by the fact that we have these short sequencing, massively parallel sequencers in play now. Now, how many people have seen an electropharogram at this point? A few people have. Okay. Often an electropharogram is going to be apparent because you've done some sort of massively parallel sequencing experiment, but you've added on a confirmation of a few questionable sequences through Sanger. So you might find that you need to be able to interpret both kinds of data. Electropharograms in general are not something that we retain when we use a short-read Illumina sequencer. They're just too much, it's way too much bulk for us to do so. So frequently what we'll store is not this electropharogram, but rather the information we derive from it. You have to pick your battles sometimes, right? If you find that storing a 1 gigabyte file for a sequencer is painful, try storing 10 gigabytes per file. It doesn't get better. All right, so this is an electropharogram, as I mentioned, and each color that you see here, black, blue, green, and red, represents a different nucleotide. So if you see at a particular location that you have a tall black peak, but there's no green peak, no red peak, no blue peak behind it, you can say, this is unambiguously a G. If you have a nice tall red, you can say, that's a T. If I have a nice tall blue, it's a C, and so on. It might seem that reading this is really, really simple. If I just sit here with I don't have a pointer stick, do I? That's okay. No, I've got this guy right here. He's now my pointer stick. Let us imagine then that we have red, black, green, 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 blue, black, green, red, etc. We can walk our way across there, no problem. And how confident are you going to feel about, let's say, this little nucleotide right here at 450? Okay, I'm claiming that this is a green peak. Therefore, I'm inferring that there's an A there. What's my chance of error associated with that? Are we in coin flip territory? Am I more confident or less confident than a coin flip? No one's really sure on this one. Okay, I'm going to claim that this position at 450 looks pretty good to me. I've got a nice strong green signal. There's very little other color that's getting a bump at that spot. I feel pretty sure that this is 450, but I need to quantify that. I need to pin it down. I'm going to argue we're much better than coin flip territory. I'm even going to say we're better than 1 in 10 chance of better than 9 in 10 of getting it right, or less than 1 in 10 of getting it wrong. But am I capable of pinning down whether that's a 1% chance of error, or a 1 in 1,000 chance of error, or a 1 in 10,000 chance of error? That's difficult, isn't it? So when we read an electropharogram, our goal is not just to come up with a sequence of letters. It's to come up with a probability of error at each of these locations. This is where the Fred algorithm comes in. We're going to spend some time talking about how that works. I want to point out some other things. I pointed at 450 because this region looks pretty good, right? But what happens when I slide to, say, this region at 720? Do you see nice and sharply defined peaks? How many A's does this long stretch, this long plateau of green constitute? Is that 4 A's? Is that 5 A's? Is it 3? We no longer have good resolution, do we, to say this peak is separate from this peak and so on. So this is something that's pretty much common to all chromatographic methods, that anytime you're doing a separation, your ability to separate these species from each other and unambiguously depends on how big they are compared to how big the difference in mass is, right? Or whatever property you're using for separation. As you're separating these nucleotides through capillary electrophoresis, your ability to tell something that's 700 nucleotides in length, apart from something with 701 nucleotides, is pretty small. Okay, likewise, look at the stuff at the beginning of the sequence. Would you feel comfortable trusting any of the base calls before 50? No, it's a big smear, it's a mess. So we have kind of a sweet spot in the middle of the electropharogram. We have problems of resolution at high base counts, and a whole bunch of smear down at the bottom. That's not a very good place to work. So being able to say which parts of this read are viable is also something we need to be able to do. So yes, we need to be able to infer a sequence from the electropharogram. That's the base calling algorithm. Fred is going to associate with each base call, with each letter, our probability of error for that letter. So how do we discern a good base call from a bad? There are several things that come into play here. So first off, we want that peak to match the beat of its neighbor. We want to see only one trace at that location, right? If let's say this is the black peak, we want there to be a black peak and no red peak and no blue peak and no green peak. We want just one color to go up and down at that location. And we want the highly probable errors to be far away. So I'm going to tell the story briefly. I've given myself a lot of slides, so I can't tell you many stories. But when I moved to Nashville in 2005, I moved to an area called 12 South. Now, at the time I moved there, 12 South was an up-and-coming neighborhood. The realtors are like very excited about up-and-coming neighborhoods. It has this, it's a neighborhood that has artists living in it, and it's got up-and-coming, and you're close to town, and so on. But it is also, what in the States gets lambasted as gentrification, which is to say that a bunch of folks could no longer pay property taxes because their house values were suddenly soaring compared to where they were living, which were often pretty lousy houses. So I moved into this place, and it was gorgeous. Gorgeous! The kitchen had been redone, nice marble top, lovely island in the middle. The floors were new and wood and gorgeous, and it was so nice. So nice. And then one day, I was wandering around in my yard, and I noticed a syringe down near the foundations of my house. And I asked my neighbors about it a little bit, and they said, "Oh yes, your house was the crack house." The house I moved into before I lived there had been the place where all the drugs were dealt in my part of the neighborhood, in the 12 South neighborhood. Some flippers had bought the house at low price, torn out the insides altogether, just left the frame there, and then rebuilt a house on that frame. But if I'd seen my house even 2 years before I bought it, I would not have felt comfortable living in that neighborhood at all. Right, so let's imagine the 12 South neighborhood then, as it was before I moved there. Imagine Dave's house is the epicenter of drug sales for 12 South, right? Now, if your house is next door to the crack house, is your property value high or low? Now, was my neighbor selling crack? My neighbor was not selling crack. They're very nice people. But they were suffering because their house was next door to the crack house. What if you were a block away from the crack house? Is your property value diminished or okay? It's still pretty bad. If you live a mile from the crack house in the very tony uphills just south of Vanderbilt University, is your property value diminished by being within a mile of a crack house? It's not. In the same way, if you're in an electropharogram and you happen to be very close to a bunch of stuff that's unresolved, that's got lousy quality, your base calling error probability is high, even if this peak looks like a very pretty peak. Proximity to something bad can tar you with the same brush, to use the phrase. Okay. Now, I've also made a lot of mention of a beat, of a particular frequency associated with those calls. I'm going to just, I want you to imagine this not as an electropharogram, but as the signal on the equalizer in your stereo when you've got the sound turned way up, right? So you're listening to something that's got a very distinctive beat. Do people listen to house music anymore, or is that a completely over thing? Maybe dubstep. How about dubstep? All right, what is the characteristic of music of the house genre or of dubstep? Just say it.
Very rhythmic.
A very rhythmic beat, okay. And by very rhythmic beat, we mean a bump, 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 bump. Okay, that is a frequency that we've got going. So I want you to imagine now that this is time, and this is the amount of sound coming out of the woofer on your stereo, right? So, womp, 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 womp. Just as is true in your dubstep music, you want these beats of the electropharogram to be spaced equally far apart. That we've got the black step, the black peak here, the blue peak here, the red peak, the green. We expect these to be spaced equally as a function of time in the capillary electrophoresis. We associate it as with a good thing if this beat is preserved, and we feel bad about it if it's not preserved. Almost any time in bioinformatics that the phrase frequency or beat shows up, it's not long before someone's going to mention Fourier Transform. Does everybody remember Fourier Transform class from any undergraduate engineering classes? Sadly, many people in our field have never taken an engineering class. And that's just sad because they have an awful lot of useful tricks for us. A Fourier transform is a way to transform an amplitude as a function of time, in this case the the fluorescent intensity as a function of capillary electrophoresis separation, to translate from this amplitude over time into a frequency signal. So if we look at this signal, we see that it's got a pretty good magnitude, about twice that of this lower signal, and it's spaced pretty far apart. So in the frequency domain, we see that we get a peak here and here to represent both the positive and negative versions of that frequency. This one has a higher frequency. You see why it crosses the zero line more times per unit, but it's at a lower magnitude. So it's going to be a smaller peak in the frequency domain, and it's going to be at a higher value. So if we saw a signal like this that represents the summation of this signal and this signal, we could decompose it to be the combination of those two frequencies. In the same way, and I have to say, Fourier transform is used bloody everywhere. If you have been listening to your iPod this morning or whatever, you've been making use of Fourier transforms. So stuff like MP3 encoding makes use of Fourier transform as a really powerful way to compress music into files. In the very old days, before we were using stuff like Fourier transform in our encoders, compressing music was a really dismal affair, I have to say. Things have gotten a lot better since MP3. So if we are looking at the signals that we see in an electropharogram, we can decompose those to a base frequency at which we expect new nucleotides to appear by using Fourier transform. Okay. Now, let us imagine that the Fourier transform has given us an expected rate at which new nucleotides are appearing. That appears along this numbered timeline at the top. Predicted location, predicted location, predicted location, predicted location. The period, the time between beats on our electropharogram is what's fallen out of the FT of the electropharogram as our set as the key rhythm at which new nucleotides are coming. So we now have predicted locations where peaks should be, and we have the observed intensity that we've observed in each channel. So when we look at a particular peak, we might find that there's a bit of shift between where we expected that peak to appear in time and where it really did. And in this case, we want to punish those nucleotides. We're going to say those nucleotides are a little misshapen. They're a little earlier or a little later than we expected them to be. As a result, we have a higher error probability associated with it. Okay. So, we now need a way to express numbers that are very, very small. As you saw from that electropharogram, most of the peaks are pretty beautiful, really. I mean, most of them are right where they expect them to be. There's only that one color and not another color at that location. Its shape is pretty nice and concave down. It looks like a nice peak, etc. So we need to be able to talk about vanishingly small differences in the error rates for these peaks. And this strategy was used in Fred, but it gets used in all kinds of different places. This negative log expression of small numbers is very, very widely used in a huge array of bioinformatics tools. So whether or not you need to ever implement your own Fred, understanding how Fred expresses those probabilities is very important to you, to understanding almost any bioinformatics algorithm. So we now have a probability of a base call error at this particular letter of sequence, and it ranges from 0 to 1. 1 meaning we're absolutely sure this is wrong, and 0 meaning that there's no chance that this is wrong. So the proximity to 0 matters quite a lot. Being 1 out of 100 is really different than being 1 out of 10,000. Could we agree that? All right. So I would point out that the Human Genome Project was funded to produce a genome sequence that was correct to less than 1 in 10,000 chance of error at each location. Right? So if the senators are only going to give you money if you produce a product that's been quantified in this way, you better have a mathematical way to talk about that. All right, so what we use then in Fred is a Q value here that represents -10 multiplied by the log base 10 of a probability. That does not inherently make a lot of sense for people, so I've given some examples here. So let's start with a probability of 0.01. This is an error probability of 0.01. Does somebody want to put that into words for us, what it means? It's not a p-value, it's a point probability. The probability of error at this base is 0.01. What's it mean? That's very good, yeah. It means that there's a 1 in 100 chance that the software has miscalled this base. Great. So probability of 0.01, if expressed in scientific notation. Everyone remember scientific notation? That's good, that's good, all right. 1 times 10 to the minus second power is 0.01. If you were talking in percentage, you would say it's 1%, a 1% chance of error. In Q, that gets expressed as 20. So 1 times 10 to the negative second, taken in log 10, is -2. -2 times -10 is 20. The math there might feel a little fuzzy, but it's going to get a lot clearer, I think, as we walk through 2 more probabilities. A 1% chance of error, a 1 in 1,000 chance of error, a 1 in 10,000 chance of error. So as we do that, we see that we go from a Q value of 20 to a Q value of 30 to a Q value of 40. So at each stage, as we get 10 times less likely to have an error, we see that the Q score, the probability, the Fred score associated with that base rises by 10 in each case. Okay, so if I report a base from my sequencer with a Fred score of 10, how do you feel about it? What's the chance of error for a Fred score of 10? 1 in... Okay, we'll work our way backward. We have a Fred score of 20 at a particular nucleotide. Do we trust the nucleotide? It's got a chance of error that's 1 in?
100.
100. Okay, we said that 20 corresponds to an error of 1 in 100. So a Fred score of 10 corresponds to an error of 1 in?
10.
1 in 10. Okay, that sucks. We do not want to deal with anything that's got a Fred score of 10. All right, generally speaking, you will find that a lot of people end up using thresholds of 30 for Fred scores in sequencing experiments, that they just trim out anything that falls below with a lower Fred score than that. Which means that they're throwing out any base calls that have a chance of error greater than 1 in 1,000. So that's pretty restricted, actually. But because we have so many sequences to work with from a modern sequencer, we can do that. All right, so we see that the number for Fred, the Fred score gets higher and higher and higher. It rises by 10 for every 10 times less likely error is. A high Phred score means a very low probability of error. And yeah, if you get below 20, don't even think about looking at that nucleotide twice. All right, when we run a sequencing experiment over at the facility, you're going to get an output to work with. And typically, that is an output in FASTQ format. FASTQ is a very widely used format, and I want to note another weird computer sciencey thing that's going on here. So if you look at the top line in this little segment of 4 lines, the FASTQ file basically digests into 4-line chunks. So this first line, starting with an @ symbol, we're not making a direct call out to somebody on Twitter. The @ symbol here means this is the address of this particular sequencing read. All right, so it's got a nice long sequence associated with it, but then here we see that in line 2, we have CCC, GCA, CTT, blah, 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 all the way down to GTT. Great. So that is the sequencing read. A FASTQ file is just a really big text file that's comprised of block after block of these 4-line segments. Each read is going to correspond to a 4-line block. So the sequence appears in number 2. The first one's just the address. The third line, typically, is just a plus symbol. This comes from the fact that we used to have a duplication of the address from the first line to tell us where this line of quality scores comes from. But in practice, it's just a plus these days. We then see a whole bunch of other letters. Now, A, what is all this business? Are those nucleotide base calls? They are not. Has anyone heard of ASCII? People have heard of ASCII. Oh, this is good. Okay, ASCII is the predominant way that we tell computers what number for a byte corresponds to a particular letter. So the letter capital A, for example, is 65 in ASCII. It's just by convention, really. The capital letter B is 66. The capital letter C is 67, and so on. All right, so these letters, in fact, represent numbers. We can store up to 255 down to 0 for an individual byte. So the A's are 65, the E's are what? Let's see, that'd be 4 more, so those would be 69. But we're not using these numbers as they are. The way that we report quality scores in FASTQs has a certain correspondence between the letter and the Phred score. Because the first letter we use is not 0 in ASCII, it's bang. So in effect, the bang represents a score of 1. And by the time you get to capital A, you're at a score of 32. So in this case, the software is saying that the first base call is C, the quality associated with it is 32. The next C corresponds to a 32, C, 32, G, 32, C, 32. This next A, however, gets a score 4 higher than that, 36. Are those acceptable base calls? For all of them, the chance of error is less than 1 in 1,000. So yeah, this is a pretty good read, in other words. When we grab these from the sequencer, the text files are absolutely massive. And you will frequently find that people have zipped them up in some way. So you might see an extension of fastq.gz. This is a zip that was created by gzip. Or you might find something like fastq.bz2, something created by blockzip. So we have lots of options for making these really large files squeeze down much better. Frequently, to shove it through a bioinformatics tool, though, you must unzip the file, which means you better have a lot of disk space ready. Okay. Graphically, one of the most common ways that we perceive this is through the FastQC software. This is created by Babraham AC UK. You can download the new version. It's free. And so when you are given a FASTQ file, if you want to evaluate whether this was a successful or a failed sequencing experiment, FastQC is your way to get there. So in this case, we are looking at quality, a box plot of quality, as a function of where in the read it is. Now remember, with old school sequencers, with Sanger's, we might have as many as 600 base calls within a read. In this case, we're using next-gen sequencers. We're using massively parallel sequencers that are generating only 40 nucleotides for each read. And you can see that at the start of the run, our median base call chance of error is above 39. So that's really bloody nice. By the time you get to the end of those 40 nucleotides, though, you've accumulated a greater and greater amount of error. And so our base call probability, our error probabilities have risen a bit. As a result, Phred score has fallen. So I'd love you to just have that relationship in mind. I mean, we talked about the math of it, but generally speaking, a 30 is a pretty typical kind of error quality, error probability associated with a base call. As errors become more likely, Phred scores fall. As errors are less likely, Phred scores rise. So that inverted relationship is something you definitely need to have in mind. FASTQC is just a way of visualizing across bazillions of reads what the error probabilities look like. All right, now we will have 5 minutes in which I rest my vocal cords slightly, and we can take quick bio breaks, but then we're right back.